project, and then Aliénor and Jan will, let's say, detail a bit more about some aspect of the project. So just to introduce myself briefly, I'm a chemist. I'm French. I have a strong interest for bioimaging, fluorescence bioimaging in particular. I've been involved strongly in France bioimaging at the level of Paris. And most of our recent activity was devoted to developing fluorescent probes and more precisely protocols for reading out those probes. So in fact, I will give a background. I don't know if you can see the title somehow, because here for me, I have this type of, yes, that's fine. So I want to introduce, in fact, our work by showing what we do with precisely fluorescent proteins. When you want to read out the signal from a fluorescent protein, most of the people will play with spectral discrimination. And then you have typically excitation and emission as discriminative channels in your system. But we are chemists, and we like to see those molecules as, let's say, sets of chemical reactions involving both ground states and excited states. And there, what we try to develop are methods in order precisely to read out with good selectivity the network. And you see that there is a topology, many rate constants, and the idea is precisely to have much more selective information by using this type of approach. In addition, if you play in the time domain somehow, or frequency domain, as you shall see, we can develop, I would say, rather simple instruments, and the hardware is not so complex very often. So what we did, just to show briefly why there is an interest there. So we developed protocols enabling, for instance, to eliminate some background interferences like ambient light or autofluorescence in the context of plant. And in particular, when using reactive molecules engaged in that case in a two-state exchange, we can, like DREAM-2, which is one reversibly photoswitchable fluorescent protein, we can easily discriminate its signal in, let's say, against the background, in that case of autofluorescence and reflection at the surface of leaves, which is what we see here, simply by using classical illumination, constant in time. There is nothing to extract selectively for DREAM-2, but when you apply some protocols, which are those protocols using modulated light, matching in terms of frequency, some, I would say, let's say, some time scale associated to the exchange, there is a way by analyzing, by Fourier transform the signal to extract selectively an information from DREAM-2. And it's something that can work also under the sun. We did experiments by labeling precisely. It's the same types of leaves from Camelina sativa. Here, under the sunlight, you have a labeled leaf here with DREAM-2. Here, it's a wild type. And we don't see any difference, basically, under such conditions with uniform illumination, constant light in time. Whereas, again, by applying this type of protocol in the frequency domain, we can extract selectively information on the labeled leaf. We use, in fact, this for various types of application. And in particular, we're interested to develop sentinel plants expressing such proteins under control of stress factors. It's something that we did. In fact, we're able to show that we could read out the extent of a stress in plants by precisely imaging selectively the level of expression of those fluorescent proteins. It works nicely, but the point is that we are using GMO, and for applications, it's not so nice. So the thought was to see whether we could, let's say by analogy, establish an analogy with the photocycle that we can find in probes like those fluorescent proteins, with something more complex, which is in that case, the whole metabolism of photosynthetic organisms. Here it's the one of Chlamydomonas reinhardtii, where we could recognize, by analogy with typically this type of network, the fact that we activate at 2 wavelengths, somehow at PS1 and PS2, and then we activate a complex photocycle containing many reactions, many rate constants. So that was somehow the idea. But here at that level, that was only an analogy of a chemist somehow. And so I asked this question to our biologist colleagues: is it possible somehow to interrogate plants with light in order to retrieve some information on their physiological state from the kinetic signature of fluorescence? And there is a long history, in fact, in that field among the biologists, but in relation to applying periodically modulated illumination, there were some works, but quite marginal and not totally analyzed, I would say. So that was really the start of our project. We discussed with the biologists, and they said that we had somehow, in order to get some selective information, to focus on a range of frequencies or time scale which was linked to the regulation of photosynthesis. It's really where they were expected to find some selective, let's say, information in relation to the physiological state from analyzing endogenous fluorescence. Okay, so that was basically the start of this project. So in the project, we have different work packages and different, let's say, tools to develop. One is, related to, I want to move this, I don't know how to do it. Up. One is related to the type of illumination applied. So as you shall see, we developed instruments for applying controlled and random light on the system at various scales. We have a part that I won't introduce, which was the development of luminescent nanosensors, but we have some work in particular at the single cell level with oxygen and with endogenous fluorescence. We have to design a miniaturized device, low cost as much as we can. There is the idea to develop a server. I will come back briefly about this in order to expand the collection of information. And then the idea was to exploit precisely this type of information such that end users, by interrogating with light their photosynthetic organisms, could receive from a server or some company that was part of the story information about what to do in order to, let's say, cure the plant or whatever. This is somehow the flow of the project. So in relation to this, we built a consortium. Here you have the names of the partners. What is also interesting is to see the type of expertise which is present in this consortium. We see that we have basically people developing instruments at ENS, so CNRS here, and Sony. We have people who are truly biologists at, in France, IBPC, at Jullien, and also at Olomouc in the Czech Republic. We have people who are really specialists of system identifications and method to analyze the signal in the frequency domain. And we have also a society dealing with many aspects of communications and then technology transfer, market analysis, and those things. So in relation to, I would say, the objectives, we had first an objective linked to sensing, really to report on the physiological state by using typically periodic excitation, cine wave or multi-cine wave, or randomly fluctuating illumination, sensors, microsystems, and then to extract precisely an information from, let's say, the modulated fluorescence arising from this type of excitation to retrieve kinetic fingerprints reporting on physiological states. There was, and there is, in fact, a development linked to a DREAM server that could be made accessible for many end users. And as a part of, let's say, exploitation of information retrieved from such experiments done in the first part here, we have a strong activity in modeling either data-based or physically meaningful somehow, or gray, mixing both. And the idea is to see whether we can then design, tailor sequences of illumination, so periodic illumination, such that using this, we could hence photosynthesis, whatever it means. So we are already almost at the end of the project. In fact, the project has started in April 22, and so we will end up in March 26. Here I listed the tasks typically, but they have been covered when I explained really what we did. Maybe a focus on the systems. We work on Chlamydomonas reinhardti as a model of photosynthesis among microalgae, otherwise Arabidopsis thaliana, and as a plant of agronomic interest, tomato. So this is basically where we are. So in fact, we collected a lot of information. We produced various things, instruments, acquisition protocols, methods for data processing. We have specific results on photosynthetic systems, but we are not biologists, the ones who you have in front of you today. And modeling as well has been a part of the story. We can make also some feedback about the difficulties that we met. And at the end, the open questions that we still have after, let's say, 3.5 years of work in the field. So now I will let the stage for Aliénor and Jan in order really to introduce some more specific development that has been made during our project. So Aliénor, the stage is yours. I just stopped to share and I propose that you share.
Can you hear me, see my screen? Okay, so thanks Ludovic for allowing us to present also today. And so I'll just explain how we came to join the project. So I've been working at Sony CSL for 6 years, and we reached out to Ludovic Jullien for my PhD as we were working on robotics for microfarms, robots in the field, and we were searching for ways to diagnose the plants and put something on them to diagnose the plants. And Ludovic had already in mind using fluorescence and light to explore plants, so it was a good combination. For today, I decided to focus on something that was a question at the beginning of the project, which is how do you calibrate light intensity for your experiment, which is quite important in photosynthetic, where the apparatus is responsive to the light level. So when you want to reproduce a protocol from the literature, and then you have your instrument, it says % on the instrument for the light intensity. How do you convert it into an information relevant to the intensity level? And then finally, it's related to imaging, is how heterogeneous your light source is at a sample. And I show you with this quick example how it's important. Here it's 2 colonies of microalgae in the squares, and we see the light, the fluorescence, their fluorescence, and the intensity is different. And for us, it's relevant if it has a physiological meaning. However, it could also come from like heterogeneity of illumination at the sample, so they receive less light, fluoresce less, or that the optics for imaging is not homogeneously imaging and creating a difference that's not physiologically relevant. And so for this, we come back to the systems that Ludovic introduced at the very beginning of the presentation, and use them not for reaction or exploration or modeling, etc. It's just we use the fact that in the reaction happening, it's a photoconversion, and when you shine light on them, they lose the fluorescence gradually. Sorry, and so the fluorescence decays with time, and so the evolution of the fluorescence carries an information which is related to the light intensity through this equation. So the rate of photoconversion is proportional to the light intensity and to absorption cross-section. And so it can be fitted with a simple mono-exponential model. And then you retrieve, this is your acquisition of fluorescence, you retrieve the tau from the fit, and then are able, from the equation, to retrieve the light intensity received by a sample. And so if you do an experiment, it would look like this. If you have your uncalibrated light source, you put your actinometer at the sample position, replace the sample by the actinometer, and then illuminate it. And this is the kind of videos you can acquire. So you see a decay with time of the fluorescence. And so this decay, if you fit it, it gives you an information on the overall light intensity if you take an average. But what you can do is fit it pixel per pixel. So for each pixel, you acquire a decay with time, retrieve the tau value, which gives you a map of tau, and then convert it to light intensity with the simple equation. And from this experiment, you can retrieve the intensity value received by the sample at each pixel. And so here is an example here. It was the image that we had on a camera. We put a film of a picture to have different light intensities and show we could sense the difference. And here it's the intensity map that we retrieve. So in micromole of photons per meter square per second. And here you see along the red line that there is a gradient at the camera, but there's actually no gradient at the image level. And so it means here it seemed like different intensity, but it's because of optical transmission of the system. So the method also allows to make this distinction, or not only have the heterogeneity, but also the distinction. And then, so we shine light on something that fluoresces and has a kinetic response. And it turns out, for the plants, it's a bit the same. When you shine light on them, the fluorescence will show some kinetic behavior with time, and there's a lot of literature about it. It's called OJIP transient. And it turns out if you fit the beginning of the curve with a mono exponential, this is logarithmic scale, well, you do it at different light intensity, retrieve the tau value for each of them. And if you fit them, you see we get a behavior that is inversely proportional to light intensity over several orders of magnitude. And so we can extract a cross-section. And so our question was, okay, it works for a leaf. Does it work for all leaves? And is the sigma value changing a lot? Or can we say there is a sigma value that allows to calibrate light intensity when I shine light on a plant? And so for this, we collected several samples of plants, measured the light intensity, extracted the sigma values from the calibration curves, and we collected samples from the Jardin des Plantes, so many different samples with different microclimates to have a good diversity. And here is the distribution of the sigma values for all the species. And we see that it ranges between 0.5 and 2 power of 6 meters square per mole. So it's quite narrow, which means we can take a value that works for all plants with a certain margin of error. And we did this in the blue light. And then we questioned, okay, but since plants absorbed in the whole spectrum, visible spectrum, will it work for other sources? And we figured out, like we tested that it was quite well following the absorption spectrum of the plant, which allows to have, like if we make the comparison, a sigma value based on the absorption absorption of plants on the visible spectrum. So we scaled it like this. And I think it's Jan's turn now.
Yeah, so let me, I will share my screen. Okay, you see it okay? Okay, thanks. Yeah, so I can maybe introduce myself first. So I'm a postdoc in Ludovic's lab, working in the frame of this DREAM project. I have a background in kind of mixed in chemical engineering and medical imaging, and now I'm pretty much just doing mostly focusing on optics, instrumental development, image processing, these kinds of things. So in the DREAM project, my main contributions are in optical instrument development, doing optical simulations also, and also development of imaging protocols. So I will just introduce two parts of my work here in Paris. So I will introduce a bit about one of the imaging protocols that we developed. And then I'll just say a few words on the macroscope system that we built. So regarding, if I can change the slides. Yep. So regarding the imaging protocols, regarding like the rationale behind why we developed these. So in this project, we are sending oscillating light to our photosynthetic species. We're recording this. We can do this if we want to do imaging, then this is quite easy to do at low speed. But the minute you go to the faster timescales, with a camera, you're limited to, I don't know, say 100 frames per second. And so if you want to image dynamics which are in the kilohertz or tens of kilohertz range, then it's very tricky. The imaging systems that you need to do this, they're complex and quite expensive. And so in the project, we wanted to go beyond just being able to image the dynamics at slower speed, but also see if we can retrieve some information at the higher end. But we needed to find some low-cost way to do this because this is in the spirit of the project. Ludovic had mentioned in the with this diagram of the DREAM project that the end goal is to have some specifications for a low-cost device, which would be at a cost not prohibitive for multiple end users, farmers and such, and researchers. So we've come up with this protocol, which essentially, with a low-cost, low-speed camera, using specific lighting techniques, we can uncover fast dynamics up to down to microsecond range using acquisitions which are less than 10 hertz. So we named these protocols REOM and EOM. They started with really looking at the theory of a system which you can photoactivate and then will deactivate, such as in the case of plants and many other photoactive systems. And what Ludovic and Tomo, who did the simulation work, had seen is that when you're sending oscillating light to this kind of system, you have many things going on. You have rate constants for the conversion between those two states, which are changing as a function of light intensity. You also have the concentrations, which are changing as a function of time, and you generate nonlinear behavior. And basically, these protocols, they exploit this, and we measure at low frequency some observable of this nonlinear behavior. And using this, we can acquire at low speed and get some information about what's going on at the higher frequency range. So looking at our first protocol, all we are doing in this first protocol is integrating over an integer number of sinusoids. So essentially, you're leaving the exposure of the camera open over some oscillations, and you collect the mean signal. And what the theory and simulations showed is that if you try at different frequencies for a photoactive system, you will have some dependence of this response with frequency. And this forms some kind of kinetic fingerprint, which has information about the dynamics at the different frequencies. With some fitting function, you can extract time constants on each pixel within your image. And so you can do imaging of fast events at low speed the long exposure may be 1 second long, for example. So you can be imaging at 0.5 hertz and you recover information that's happening in the microsecond. So that's the REOM protocol. Next protocol, a little bit different. So instead of just sending one frequency, we send a second frequency, but 1 hertz different from the first. And what this does, if we look, it's maybe easier to look in the frequency domain, which we see here. So we have one frequency, we have another frequency, and we have the camera frequency. So we cannot measure these frequencies with our camera. But when we send this kind of lighting protocol to our system, we generate terms at the addition of these two frequencies, but also at the difference. And this difference, if we set this at 1 hertz, we can collect our signal, not at 1 hertz, but you can collect a 1 hertz signal at low speed, 30 hertz, for example. And if you shift these frequencies and you test along, this difference remains the same at 1 hertz. So you keep measuring at 1 hertz, making a fit on each pixel. With this, you can then extract the time constants using this kinetic fingerprint that we've obtained. And so this is the EOM protocol. So once the theory and simulations had been kind of concluded, we validated these in known systems. So using the system that Aliénor had mentioned, this DREAM-2 photoswitchable protein. So this is the work of Hélène Merceron. So she basically tested these protocols to see that they work at the millisecond scale with this protein. She put the protein into or she labeled cells with this protein. She ran the HEOM and REOM protocols. If you look at the average curves, she has the response that we expected, and fitting on each pixel, she has a map of the dynamics of the exchange. She also then tested to see if we could go down to the microsecond scale. So she used beads labeled with a palladium complex in this case. She ran the REOM protocol on this, and we got time constants which were in the microsecond and the same as what we expected from traditional like accepted techniques. So that was nice. These were validated. Then we thought, what would happen if we tried this with a leaf? So we ran this protocol, the REOM protocol in this case, on a leaf, which was not treated with anything, so just taken from the garden and popped in the macroscope. And as a function of frequency, this is the REOM response that we received. Then we also tried with a leaf treated with DCMU it's an inhibitor of photosynthesis, just to see, can we see a difference in the kinetic response? And sure enough, we've seen a difference in the response. You see that the curves don't look like what we had before for fitting. So in this case, we're not at the stage of fitting and recovering time constants. So for now, to create images, we're just taking the ratio of the REOM signal at low frequency over high frequency, so we can generate some kind of map of this REOM response. So using this, we did some experiment to see if we could follow the uptake of DCMU into a small plant. So we just applied DCMU to the roots of a small plant. We applied the REOM protocol at different times, up to almost a day, and we could see quite nice selective imaging of the location of the DCMU within the small plant. So that's kind of as far as we got with those protocols. So going from there, so I will just mention a few things about the macroscope instrument that, well, these plant experiments were done on. So this is how the system looks. You have a bunch of optical components. Essentially, there's an illumination system, and you have an imaging system in here, and you have loads of parts to keep them all in place. It images over a 1 centimeter by 1 centimeter area, so we can work with a number of different kinds of samples. We work with solutions of protein on slides. We work with leaves, also small plants in pots also. Illumination wavelengths, you have a load of different choices to choose from, so we can work with many different systems. For the dynamic light that we apply to these systems, we can go for, we can send light over a big range of frequencies. So from 100 kilohertz down to 10 to minus 3 hertz. We can acquire at 100 frames per second on the camera. And if we want to not have imaging, but go much faster, we can go up to 1 megahertz on point detection mode. So that's one of a family of instruments that we've built at ENS. So I think Aliénor is going to speak on the next slide to mention about them all.
Yeah, so there was Jan's microscope, and then we also wanted to run parallelized experiments. So the strategy was to work with microalgae instead of plants to acquire a lot of samples with this instrument, where we also decide on the protocol that we apply. And then to test about miniaturization and low cost, we try to reproduce a given experiment with this instrument on a 3D printed microscope and manage. So we showed that when you know exactly where you want to go, you can really reduce the cost of a device. And to push it further, we wanted to think again about parallelized experiments, but for plants. And for this, we fell upon a work where a 3D printer was transformed into an automated robot for duckweed analysis. And so we designed a new tool that would go on the robot, and it allows to measure fluorescence. Here it's point detection, but we can replace it by a camera. And this one holds in your hand, or actually in the robot. Can you go to the next slide, Jan, please? So this robot is called a DuckBot, and it has several tools to prepare samples, to prepare solutions, to take images, etc. And we decided to add the fluorescence measurement. And now we are working, if you can show the next, on a fully automated platform where we would also control the growth conditions, even by applying different light conditions to the different wells of the microplate. It's still under development, but we are happy to discuss about it if it can be interesting. It's so during the growth, it's on the middle, and when you want to do an experiment, it lifts up, picks up a tool and does the measurements. Okay.
So let's conclude. Thank you.
Thank you.
Thank you.